unofficially welcome you all to today's program, that is International Lecture Workshop on DNA Damage and Repairing Human Health, Scientific Breakthrough in Indian Context. So today we have with us Dr. Muralidhar L. Hegde, Professor and Director, Division of DNA Repair Research, Houston Methodist Research Institute, Texas, USA. We have virtually with us Dr. K.S. Jagannath Rao, Pro-Chancellor, KL Dean to the University, Vijayawada. Now I request our Chairman, Dr. M.S. Mustaq, to escort the guest and President of the program onto the stage. Thank you, sir. When the letters are combined meaningfully, they form words. When the words are combined meaningfully, they form sentences. With the addition of tone of music into this, it becomes lyrics. With the addition of voice of expression, it becomes a song. With the addition of emotion of unity, it becomes an anthem. Now I request Anupama and Suma Kote to ignite a spark of positivity in this hall through the college anthem.
ನವೆ ಬೆಳಕು ಬೆಳಗೋಕೆ ಬದುಕು ಜ್ಞಾನವೇ ಬೆಳಕು ಬೆಳಗೋಕೆ ಬದುಕು ಎರೆಯುತ್ತ ಜ್ಞಾನ ತೆರೆಸುತ್ತ ಕಣ್ಣ ಈ ವಿದ್ಯೆ ಹೊನ್ನ ಕಿರಣ ದಿನಪಾಠ ಪಠಣ ಗುರು ಬುದ್ಧಿ ವಚನ ಇದು ವಿದ್ಯಾದಾನ ತಾಣ ಪಡೆದಾಗಲೇನೆ ಸಿರಿ ಶಿಕ್ಷಣ ನಮ್ಮ ಅಂಧಕಾರ ದಮನ ಮಂಗಳೂರು ವಿಶ್ವವಿದ್ಯಾನಿಲಯ ಮಂಗಳೂರು ವಿಶ್ವವಿದ್ಯಾನಿಲಯ ನಮನ ನಮನ ಜ್ಞಾನವೇ ಬೆಳಕು ಬೆಳಗೋಕೆ ಬದುಕು ಜ್ಞಾನ ದೇಗುಲವೇ ಬೀಡು ಬಿಟ್ಟಿದೆ ಕಡಲಿನ ತಡಿಯಲ್ಲಿ ಅರಿವು ಮೂಡಿಸುವ ಗುರುವೇ ದೇವರು ನಮ್ಮ ಈ ಗುಡಿಯಲ್ಲಿ ಎಲ್ಲ ಸಾಧನೆಯ ಮುಟ್ಟೋ ಮೆಟ್ಟಿಲು ಹತ್ತೋದಿಲ್ಲೆ ಮೊದಲು ವಿದ್ಯೆ ನಮ್ಮಯ ಹಕ್ಕು ನಾಳೆಯ ದಿಕ್ಕು ಜೀವನದ ಸೆಲೆಯೂ ಜ್ಞಾನವೇ ಬೆಳಕು ಬೆಳಗೋಕೆ ಬದುಕು ಎರೆಯುತ್ತ ಜ್ಞಾನ ತೆರೆಸುತ್ತ ಕಣ್ಣ ಈ ವಿದ್ಯೆ ಹೊನ್ನ ಕಿರಣ ದಿನಪಾಠ ಪಠಣ ಗುರು ಬುದ್ಧಿ ವಚನ ಇದು ವಿದ್ಯಾದಾನ ತಾಣ ಪಡೆದಾಗಲೇನೆ ಸಿರಿ ಶಿಕ್ಷಣ ನಮ್ಮ ಅಂಧಕಾರ ದಮನ ಮಂಗಳೂರು ವಿಶ್ವವಿದ್ಯಾನಿಲಯ ಮಂಗಳೂರು ವಿಶ್ವವಿದ್ಯಾನಿಲಯ ನಮನ ನಮನ.
We are immensely grateful for all of your presence here today. And now, it is time to express it through words. I request our respected Chairman of Applied Zoology, Dr. M.S. Mustaq, to welcome the gathering.
Good morning to one and all present here. Our esteemed chief guest, Dr. K.S. Jagannath Rao, Pro-Chancellor, KL University, Vijayawada, Andhra Pradesh. Because of some reason, medical emergency, he could not come in person here, but he is there with us. Virtually, he will be presenting his talk. So I am also grateful to him for coming in virtually. And guest of honor, Dr. Muralidhar L. Hegde, Professor and Director, Department of Neurosurgery, Center for Neurodegeneration at Houston Methodist Research Institute, USA. Our beloved Vice Chancellor and President of today's program, Professor P. Subramania Edipadi-Thaya, esteemed colleagues, research scholars, my dear students, supporting staff members, friends from media, invitees, ladies and gentlemen. I extend a very warm, cordial welcome to one and all. Present here, I am greatly honored to address distinguished gathering of the International Lecture Workshop on DNA Damage and Repair in Human Health, Scientific Breakthrough in Indian Context. On account of Azadi Ka Amrit Mahotsav, an initiative of the Government of India to celebrate and commemorate 75 years of independence and the glorious history of its culture, people, and their achievements. Before I introduce dignitaries on the dais, let me brief about the department and the conference. The Department of Applied Zoology is started as a division offering MSc in Applied Zoology in the year 1988 and Zoology in the recent year 2018. This developed into full-fledged department in the year 1994. The founder chairman of the department, Professor S.N. Hegde, served the department till 1997, until he was elevated to the position of Vice Chancellor, University of Mysore. Department presently has 4 faculty members with expertise in the areas of genetic toxicology, genetics and toxicology, neurobiology, population genetics and genomics, radiation biology, physiology, cytogenetics, fishery biology, molecular biology, biodiversity, which are the areas of research of the department. The DBT Ramalinga Swamy Fellow is also working in our department. The faculty has received fellowships from Commonwealth grants, NCERC, Canada, AICT, DAD, and DBT. Presently, there are 85 students pursuing their MSc and 24 research scholars working for their PhD degree. Subject taught for postgraduate students include several applied aspects of zoology in keeping with the recent development in animal sciences. Our students have performed well and now placed in national laboratories in India and abroad, like Australia, Germany, Sweden, Singapore, Japan, Taiwan, and USA. A special paper on wildlife conservation and management was introduced in 1994 with special assistant from UGC, New Delhi. The department was supported by DST FIS funds in 2005. The inducted into UGC SAP during 2009, we are now elevated to DRS level 2 from 2015. Department has received DSIR infrastructure funding from UGC along with RFSMS and BSR fellowships. The research endeavors of the department have been funded by various agencies like DST, SERP, DBT, AICT, and BRNS. DST also has awarded Young Scientist Fellowship in the department. The department is actively involved in the research area of biodiversity, conservation, and projects under this have been funded by MacArthur Foundation, New York Wildlife Society. One of the special features of the department in the Molluscan Shell Museum, which houses collection of shells, more than 2,000 molluscan species from all over the world. It has been housed in exclusive museum with grants from UGC New Delhi. The museum also has collection of regional fauna. The promote interaction among academicians, students, and department has organized several seminars, symposia, workshops sponsored by different agencies. Department has National History Association. It arranges lectures, quiz program, photography competition, field visit, tour to promote excellence in teaching and learning. Fruits of teaching and research should benefit the society. To inspire in the students, department was arranging extension activities. Faculty of the departments are involved in People Biodiversity Registry, the government of Karnataka, and have thus contributed recording fauna of local village. Department also celebrate Wildlife Week through exhibitions, street play, competition, etc. to sensitize the public on importance of wildlife and biodiversity. Department is organizing every year campus bird count and Wild Bird Day since 2016. Today, Mangalore University, Department of Applied Zoology, under the leadership of Professor Subramania Adipaditaya, is organizing one-day international lecture workshop on DNA damage and repair in human health, scientific breakthrough in Indian context. As a part of celebrations of 75 years of India's independence, Bharat Ka Amrut Mahotsav. India's progress in the field of science and technology has been exponential since our independence. And research and development work has addressed many of the problems of society is facing now. Science is now regarded as one of the most powerful instruments of the growth and development. India is ranked 6th globally in the number of scientific publications and 9th in the number of patent field. India is recognized globally for its scientific rigor and potential. After all, this is the land of Ayurveda, the land of climate sensitivity demonstrated through the Chipko movement back in 1970s. Land where successful nuclear tests like Pokhran II were conducted. the land where science maestros Sir C.V. Raman, Nobel Prize in Physics 1930, and Anna Mani were born. India was able to gain pace in scientific discovery only after independence, getting back in the reins of democracy and planning the years ahead to regain our prosperity and encouraged people to work towards scientific and technological advancement. In all these years, India witnessed massive shift in the field of discovery by strategically aligning its skills and resources. Few glimpses of accomplishment of India in the field of science and technology post-independence and developments were progressing towards. For example, 1947 to 57, establishment of 5-year plan which prioritized scientific research. Being the first plan, it prioritized laying foundation of scientific research in the country, largely focused on initiatives of building or enhanced national laboratories and research center. It recognizes 11 research institutes at the national level and stressed their importance in the development of country's future. For example, National Physical Laboratory of India, New Delhi, National Chemical Laboratory, Pune, Central Electrochemical Institute, like that, 11 research institutes were formed. In 1957 to 1967, it was focused on agricultural research and emergence of green revolution. In 1967 to 1997, Aryabhatta, India's first satellite, was launched. In 1977 to 1987, Agni, India's missile, were launched. In 1997, in 1997, in that period, DNA fingerprinting in India came to existence in 1988 when CSIR CCMB scientist Dr. Lalji Singh developed the technique and made it available for use, making India the third country to develop its own DNA fingerprinting probe. In 1997 to 2007, Pokhran 2 nuclear test was conducted. And 2007 to 2017, Chandrayaan-1 mission to the moon. Polio-free India. India received polio-free certificate from the WHO on 27th March 2014. Mars orbital mission, also known as Mangalayaan, engagement of startups were done. And 2017 till present, ISRO's Gaganyaan program is launched. And to COVID-19 vaccine research and vaccination drive, all you know how many vaccines we have contributed to different countries. Therefore, today's program will enlighten the students and my fellow mates very interesting topics on the field about aspects of DNA damage and repair in human health, and fill you with the knowledge of emerging field in the biology that has various application. We have received more than 400 registration participants, and this program have 2 lectures. After this, Muralidhar L. Hegde and Jagannath Rao will be virtually, he will be talking to us. And after that, we have we have photo quiz context on biological science based on the Azadi Ka Amrit Mahotsav, and we have afterwards cultural program. Now, we have as 3 eminent dynamic personalities who are immense glory of today's conference. Being the middle class family with their hard struggle in their early days, now they achieve topmost post, doing good work and making our country proud. Today we are very proud to have them with us to commemorate the 75th independence of India, Azadi Ka Amrit Mahotsav. The convener of this program, as a convener of this program, it is my proud privilege to welcome our chief guest. He is online, K.S. Jagannath Rao, Pro-Chancellor, KL Deemed University in India. He currently Pro-Chancellor, KL Deemed University, India, and involved in the transformation of university into global model. He has obtained PhD degree from Department of Zoology, Sri Venkateshwara University, Tirupati, India. From then, he has vast professional work experience and worked different national and international institute. Name few: CFT Rai Mysore, University of Virginia, Medical School, Charlottesville, USA, Medical University of South Carolina, USA, Methodist Research Institute, Houston, Texas, USA. He has good experience in institution building, experience in served as a CEO, director, and distinguished professor of Inticas at AIP in Republic of Panama. His advocacy has resulted in the construction of $30 million research facility known as PRISM, completed in 2014. He has his credit many awards, academic membership. He has published 165 publications, many books, and he has guided 16 PhD students. Sir, your august presence empowers our students, faculty present here to carry out research in frontline and emerging areas. I welcome you, sir. We are honored with the presence of Dr. Muralidhar L. Hegde as a professor of Department of Neurosurgery and Center for Neurodegeneration at Houston Methodist Research Institute, USA. Dr. Hegde directs a research program focused on delineating a molecular insight into involvement of genome damage and repair response in human brain pathologies and developing novel mechanism-based treatment strategies for this disease via neurodegeneration, primarily for motor neuron disease. To support Hegde's growing successful research program, Houston Methodist leadership created a new division of DNA repair and appointed him as a director in January 2022. Hegde Laboratories specializes in the state-of-art molecular assays for genomic instability and damage and repair, utilizes CRISPR-Cas9 technology, human patient-derived induced pluripotent cell, novel mouse model and human tissues. Dr. Hegde earned his PhD in biochemistry and neuroscience from University of Mysore, India in 2006. He performed his graduate research in Max Planck Institute of Biophysical Chemistry, Göttingen, Germany, as DST DAD visiting fellow. He was assistant professor at University of Texas Medical Branch, UTMB, Galveston, Texas, before joining HMRI in 2013. Dr. Hegde's research has been continuously funded by multiple grants from National Institute of Health and other foundations. Currently, Hegde Lab is supported by 4 NIH grants and industry grant. Recently completed external grant include awards from the American Parkinson's Disease Association, Alzheimer's Association, ALS Association, Muscular Dystrophy Association, Mueller Foundation. Dr. Hegde has published nearly 100 peer-reviewed publications as well as 10 book chapters and fundamental review articles. These articles have received more than 5,000 citations. H-index is 35. Recent publications include Nature Communication, PNAS, ACS, Nano, and JBC. He has served several grant review boards, like example USA Alzheimer's Association, French scientific grants, in addition to study sections of NIH and Department of Defense. Further, Dr. Hegde is Deputy Chief Editor of Metabolic Brain Disease and Associate Editor of Journal of Alzheimer's Disease. He has also served on various editorial boards, half-dozen journals. Hegde Laboratory has been funded by 4 National Institute of Health, NIH, R-level grants, and grants from American Parkinson's Disease Association, Alzheimer's Association, ALS Association, Muscular Dystrophy Association, and Mueller Foundation. With this brief introduction, I would like to request our Honorable Vice Chancellor, formally welcome Dr. Muralidhar L. Hegde. Thank you. With this brief introduction about the dignitaries, let me have our honor of introducing President of today's function, Professor P. Subramania Edipaditaya, Honorable Vice Chancellor of Mangalore University, to this august gathering. Professor Edipaditaya is well known to all of us and requires no introduction. He is a guide and backbone of all our activities on the campus. As soon as assumed Chancellor of this university, he has initiated several steps to bring out several academic and administrative reforms in the university. He has been relentless in promoting service, quality, integrity, and sustainability, and cornerstones of our institutional leadership. For the benefit of young friend here, let me present very brief resume of our Honorable Vice Chancellor. Dr. P. Subramania Edipaditaya is a native of Kokkada, Dakshina Kannada district. He is currently 9th Vice Chancellor of Mangalore University. He graduated from first batch of Postgraduate Department of Commerce, securing first rank and gold medal, MCOM 1982, from Mangalore University. He also completed his doctorate in consumer behavior from the same university in the year 1992. He did his postdoctoral research from Nottingham Business School, Nottingham Trent University, England during 2001-2002 under the Commonwealth Fellowship Scheme. He is currently a member of various committees, like state universities, for drafting uniform calendar of academic events appointed by Karnataka State Higher Education Council, Government of Karnataka. He is also a member of committee to revamp evaluation system in higher education in the state, appointed by Karnataka State Higher Education Council, Government of Karnataka. He also on the board of management of Karnataka State Open University. He worked as an assistant professor and trainer and consultant at National Institute of Training for Industrial Engineering, Mumbai during 94-95. He has published more than 50 research papers in national and international journal of repute. He has successfully supervised 23 PhD scholars in the area of resource management, HRM, and human resource development, HRD, and marketing during his 4 decades of postgraduate teaching and research experience. He is also a proud recipient of Elan International Award 2003 from Scotland, UK, and Devang Mehta National Award for Best Teacher in HRM in 2009. Dr. R. Sudarshan Memorial Lifetime Achievement Award for National Achievers, Administration and Education in 2020. He has served as a finance officer, registrar evaluation, registrar of Mangalore University, gathering the administrative experience nearly 10 years. We are immensely honored by your presence, sir. I request Professor Bhaskar Shane to florally welcome Professor P. Edipaditaya, Honorable Vice Chancellor. Now I welcome all the participants, those who have come from in and around Mangalore, chairmen of various departments, faculty members, research scholars, students, non-teaching staffs, media friends, and invitees. Thank you, one and all. Thank you. Jai Hind.
Gratitudes overloaded, sir. The warm, bright glow emitted from Diya is considered auspicious. It represents enlightenment. It represents prosperity. It represents wisdom and knowledge. Diyas represent the triumph of good over evil, light over dark. Moreover, it is our Indian culture. So now I request Professor Muralidhar L. Hegde to inaugurate the ceremony by lighting Diya. I request our President of the program and Chairman of the department to join the L. Hegde to embrace the audience through his inaugural speech.
Good morning, everyone. Respected Vice Chancellor, Professor Subramania, my friend, Professor Mushtaq, Professor Sripada, who I know very well. I visited here when he was the chair a few years back, and Professor Bhaskar Shanoy, and all the leadership in the university. And one of the favorite part of me visiting here again is to meet and interact with the students. I had a great time and great interaction with the students last time when I came here. So all the faculty and students from Mangalore University. So as I mentioned, I am really delighted to be back in Mangalore University after a gap of 4 years. And I am super excited to inaugurate this workshop on an important topic of DNA damage repair in human health, and particularly in Indian context. And I really feel that, this is a very appropriate topic and very timely. And as we celebrate, this is on the occasion of 75th year of India's independence, or Amrita Mahotsav, as we, pay homage to our freedom fighters, nation builders, farmers, and innovators. If you look at the field of DNA repair in the last, since Watson and Crick discovered the structure of DNA in the late 1950s, there are immense, contribution from Indian scientists and Indian origin scientists, which kind of gave new direction to the field of DNA at different stages. So, since, Watson and Crick discovered the DNA structure, it was kind of carried for next 2 decades by Professor Vishwamitra from Indian Institute of Science, who kind of discovered different structures of DNA, like zDNA, or different, helical structures like bcDNA, and their importance or their functional significance. And we were lucky, both me and Mushtaq worked as colleagues during PhD in Mysore, and also we spent some time in Germany together. So we got the opportunity to work in Dr. Vishwamitra's lab when he was alive. He passed away in 2001. And we got to know the immense passion Vishwamitra had to understand, different structures of DNA in terms of DNA health and disease. And, many of you may know, he at one time from, I think, 1976 to 1984, he published about 14 papers in Nature. And one issue of Nature in 1976 has 3 papers from his lab. A single issue had 3 papers. And actually, we used to call him, like, if Watson and Crick were to be considered as Brahma of DNA structure, Vishwamitra is like Vishnu, continued the field forward. And, if you come forward, like Dr. Mushtaq mentioned about Lalji Singh, discovered DNA fingerprinting technique, and Hargovind Khorana, Indian origin scientist, who discovered the DNA, nucleic acids and protein synthesis, nucleic acid codes, and so on. And Professor Jagannath Rao, and I don't know whether he's listening, who was supposed to be here, unfortunately for some medical condition of his wife, he could not come, but I'm sure he will be giving a talk. So he was one person, there are, I may miss some names, but I'm trying to mention some names who have carried the field forward in, more than a decade. So he was one person who kind of studied the DNA damage in human diseases, particularly in brain diseases. So DNA damage till then was kind of studied for, people knew that DNA damages, but it gets repaired. So it was mostly studied for cancer, therapy, any chemotherapeutic agents or radiation that damages DNA sufficiently to kill the cells. So it was mostly studied for that purpose. So Jagannath Rao was the one who initiated the study of understanding of DNA damage in normal health, during aging process as well as human diseases. And both me and Dr. Mushtaq got the opportunity to do PhD with him, and we got a lot of opportunity to interact. That was the time that field was sprouting everywhere all over the world. We got to interact with a lot of other groups working in the area in different countries, and we visited Germany in one of the leading labs in Max Planck Institute, Thomas Jovin's lab, to do some DNA damage studies, where we looked into neurodegeneration associated factors, how they affect DNA damage. And since I completed PhD, I got another opportunity to work with Professor Mitra. And some of you must have met him. He visited here about, I think, in 2018, late 2018, and gave a talk here. So he was one of the leading DNA repair scientists in the United States. So, we are talking about DNA structure and DNA damage, and DNA repair came into prominence in, late 1990s and early 2000s, and that was the time many of the DNA repair enzymes were discovered. Otherwise we knew only in bacteria you need about 4 enzymes to repair most of the DNA damages. When it comes to human beings, these are so complex that these are linked to different processes. The cell division, stress conditions, transcription, these are kind of co-opting with different machinery. So Dr. Mitra, in fact, in early 2000s, he discovered 4 new DNA repair proteins which are involved in oxidative DNA damage repair. So he was one of the leading scientists in understanding how DNA is being repaired and what are the implications of repair defects in human diseases. So, I just try to remember few scientists of India, from India and Indian origin scientists who contributed to this field. And more recently, in the last 10 years, the field has emerged. And, I should also mention that, DNA is important for, every cell's function, but the field has advanced in terms of using DNA in various technologies, in research methodologies like, even drug delivery, vaccine development, and so on. So it's also, the DNA field has developed in terms of technologies. And more recently, we are trying to understand how DNA can be target for delayed, preventing or delayed aging as well as maintaining human health for a long period of time. And this is particularly important for cells which live longer in our body, and they are neurons. Most of the brain cells or central nervous system cells we have in the body, about 99% of them we have when we are 3 years old, and then they live, till we live, so about 70 to 80 years. So it's very important to maintain, DNA damage and DNA health during aging and human diseases. And now we are, in the last 10 years, the field has emerged to see how DNA can be targeted to prevent many of these diseases. I think this workshop will give a great opportunity for the students to learn a little bit of history of DNA damage and repair, as well as the emerging science about DNA and the future, future prospects for the next 10 years, how the field is moving. So as I mentioned, I am really excited to be part of this workshop, and I thank my friend Dr. Mushtaq for selecting the topic and organizing this workshop, and the Honorable Vice Chancellor and all the leadership of the university for blessing him and supporting him to conduct this workshop. And I'm really looking forward to both mine and Dr. Jagannath Rao's presentations as well as interaction with all of you. Thank you very much.
Thank you very much, sir. Now, the Honorable Chief Guest for today's event, Dr. K.S. Jagannath Rao, will virtually address the gathering.
Good morning to all of you. I missed the occasion of the emergency happening here. I really apologize, Vice Chancellor, for I'm not going to be, I was not a great opportunity to meet you, sir, and meet all the faculty and students in the university. I'm delighted to, give a few words of inaugural things. I'm very happy to announce that Dr. Muralidhar Hegde and Dr. Mushtaq Mohammed, who are my former students, my colleagues, did a fantastic job in Indian brain science. Especially, I would like to cite two things. Dr. Mushtaq Mohammed was the first one to build a depression brain bank in Mysore, ever first done in India. And Dr. Muralidhar Hegde was responsible in supporting to build a Parkinson bank in Nimai. So these are the two people, made an international collaborations, also interacted with a number of scientists, especially medical doctors, leading to new inventions in Indian science. And today I'm very proud that Dr. Muralidhar Hegde has become a leader, and he is heading a center, DNA repair center in US, especially focusing on very important part in the brain. Why brain? Around the world is cancer or is the brain? In the case of cancer, targeting has become a challenge. Understanding metabolic targets has been a challenge. But in human brain, understanding human brain itself has become a challenge. We only understand 50% of human brain. Only 10% of human brain. There is a huge potential to work on that. And me and Dr. Muralidhar Hegde will be very happy, along with other scientists from around the world, to support Mangalore University. Now, if you people build a center on this thing, then there is a huge amount of internship to the students. Also, go to support students, there's a lot of money available. So we very happy to I to make sure. I also remember the Bangalore University when I was there, registered for PhD in neuroscience. That was my first to Bangalore. You know, these things and chancellor for now up to in things for the from US, you know, with to make different Indian science. Thank you very much.
Thank you, sir. To a normal person, every colorful butterfly is just a butterfly. But for a zoologist, it can be a moth or it can be a butterfly. To a normal person, it is just the cheetah. But for a zoologist, it can be a cheetah or it can be a leopard. This is where a zoologist stands different. Being a zoology student, it's our proud to say that we have made a campus wildlife video. Now, I request the president of the program, Professor P. Subramanya Edapaditaya, to distribute the CDs of campus wildlife video. Please, sir. Thank you so much, sir. A leader is the one who knows the way, goes the way, shows the way. I'm happy to say that we, the Mangalore University people, have such a leader, such a strength with us. It is none other than the president of today's program, Vice Chancellor of Mangalore University, Professor P. Subramanya Edapaditaya. Sir, I request you to give a presidential address.
Esteemed Dr. Muralidhar L. Hegde, Professor, Houston Methodist Research Institute, Texas, USA, who just inaugurated this international lecture workshop on DNA damage and repair in human health, scientific breakthrough in Indian context by providing a thumbnail sketch. The esteemed chief guest, Dr. K.S. Jagannath Rao, Pro-Chancellor, KL deemed to be University Vijayawada, who joined us through virtual mode and also explained the scope for DNA research and DNA repair pathways and so on and so forth. Convener and Chairman of the Department of Applied Zoology, Dr. Muhammad S. Mustak, distinguished faculty members, young student friends from not only from the Department of Applied Zoology, but also other sister departments like Biosciences, friends from media, and ladies and gentlemen. In any airport, you will come across two gates. One is the arrival gate, the other one is the departure gate. We are waiting at the departure gate. I am 63 years old. But you, youngsters, you are at the arrival gate. The point to be noted here is, health is wealth is not only an old adage, but it is the real fact of life. Only when the health is lost, we try to understand and appreciate the value of health. So that being the case, normally when I interacted with my one senior professor, Dr. Chandrasekhar Shetty, the other day in Hotel Woodlands, we were talking about health disorders, health-related issues and all. In his own typical style, he told me, this human system, biological system, is highly complex. I pondered over this matter, and I also realized it is neither a child's play nor Sunday gardening to understand the complexities of human physiology, the system the processes. So it means I need not be a student of bioscience or applied zoology to understand and appreciate this complexity. I am also having working knowledge about DNA damage, DNA repair, and DNA repair pathways. So many things. Out of my own curiosity, I have started reading certain literature relating to that area. So that being the case, in my own style, because I belong to the field of human resource development, and I will always confess that human development should always precede human resource development. So human development is the pivot around which all other economic, social, political, sociological activities rotate. So when we talk about human development, invariably, three things are very, very important. One is predictive maintenance of the entire system, preventive maintenance. Prevention is always better than cure. The other one is productive maintenance. Because you're inviting problems and coming to grief later on. No. Understand the health damages, health-related issues, and in what way we can maintain our system in a very, very productive and useful manner. Maybe our food habits, or exercise, or exposure to ultraviolet sun rays, which is subject to irradiation, and we are likely to contact what you call skin cancer, so many things. So that being the case, predictive maintenance means, look here, this is the signaling system. You are not all right. Because as I understand, particularly the mental neurological disorders, MND we call it, and the genes, the cells, after some age, because due to the factor of aging, they fail to regenerate. There is a reason as to why the dementia or Alzheimer's disease, when I was in UK, I was interacting with a lady who is suffering from Alzheimer's disease. Every day she is asking me, what day is today, Subra? What's the time now? After 10 minutes again, I used to get the call. So it means it's a serious concern for every one of us. So there are several concerns, issues, and apprehensions, and also challenges to combat all these things. So that being the point, we understand that, scientists discover new mechanisms cells use to fix damaged DNA. In the UK, in the US, that is what Dr. M.L. Hegde tried to impress upon all of us. There may be several causes for disease and medical issues. The primary cause could be DNA damage, and it may be due to endogenous or exogenous agents. And the entire problem is, remember, linked to aging, the process of aging, then cancer and motor neuron disease, MND. As a common man, I don't belong to the field of science, but I operate in the applied science field, the social science, and I deal with the human resource development. Therefore, it's a serious matter of concern for me in all endeavors. So that being the case, we cannot stop the factor of aging, but we can only delay the factor of aging. So it means we have got a new toolkit, that is what people say. New toolkit for understanding DNA breakage and exploring DNA pathways, repair pathways. But the matter is very, very serious and important, because in the scientific literature, I could grasp certain things like failure to fix DNA breaks in our genome can impact our ability to enjoy a healthy life at an old age. I'm entering to that. Because I'll call myself as an old man only after attaining the age of 80 years. I'm fully confident. So at an old age, as well as leave us vulnerable to neurological diseases like MND. This neurological system, particularly in the brain, it's too complex. Dr. Devadiga, I used to interact with him. He used to tell me, 80% I know, 20% Kuda hai to bala karega, inshallah. With that kind of spirit, they'll go and operate upon a patient. And he says, more often than not, operation has become successful, patient is dead. Not because of my lack of knowledge or lack of skills or lack of competencies, but 20% is still unknown. So is the case with Dr. Dev Prasad Shetty. So Dr. Dev Shetty, you know it, before going to the operation theatre, he will offer prayers. So 20% is very, very important. It has become a very, very strange puzzle. So ultimately what we can do, together we can. It means collaborate to innovate is the order of the day. Together we can. So collaborative research assumes a lot of significance in the current context. We have got H index of 65 or so. And our teaching fraternity is contributing to this kind of research, innovation and excellence in Mangalore University to a very significant extent. But what I'm trying to impress upon is there should not be any repetitive research. It should be refutative research which will question and challenge the false dogmas. That will happen because earlier we were talking about publish or perish. Later on we say innovate or perish. So much more than the publications, we should try for patents. That should be the route to be chosen by all of us. And ultimately we are concerned with the genome instability. Because normally we say human behavior is highly difficult to understand, predict and control. But nowhere it is said that it is impossible to understand, predict and control human behavior. In spite of genome instability, we can definitely attempt to that only through collaborative research. So routinely used biological, physical and chemical agents impact human health. The research findings will tell us. Testing their genotoxicity and regulating their use have become very important today. More important today than ever before. So it means ultimately the punchline or the bottom line is preserving genomic sequence. Information in living organisms is important for the perpetuation of life. So all said and done, it's a complex system. Ultraviolet light and DNA damage. Best example could be exposure to solar ultraviolet irradiation, cancer, that kind of association. So it means this one day international lecture workshop will definitely throw sufficient light on all these aspects. But my fundamental question as a man on the street or a layman is, can we prevent DNA damage? If so, to what extent? It means causes, consequences, cures, remedial measures, so many things. And we'll be reminded of again, to come to the starting point, predictive maintenance, preventive maintenance, and productive maintenance will be very, very crucial, all of us. I congratulate Dr. Muhammad S. Mustaq and his team for organizing this international lecture workshop on DNA damage and repair in human health, scientific breakthrough in Indian context. Try to make the best use of every moment of truth available through interactions and deliberations in this, because you're dealing with the stalwarts. You'll be definitely enlightened, because Dr. Muralidhar L. Hegde, he is a promising person in this field, DNA damage and repair and other things. And teacher of the teacher and the researcher of the researchers, Dr. K.S. Jagannath Rao, is also contributing through virtual mode. So these are all brilliant brains available for us, and it is up to you. You are holding the bird. You can save the bird or you can kill the bird. You are holding the bird in your hand. The answer is in your hand. Best wishes and greetings from Mangalore University. Thank you very much for your time and opportunity. Jai Hind, Jai Karnataka.
Thank you very much, sir, for your informative and wise words. Thank you is not just a word. It is a word of gratitude given to all those strong pillars behind the success of the program. Now, I would like to invite our respected professor, Dr. K. Bhaskar Shanai, to propose a vote of thanks.
Good morning to all. Respected dignitaries on the dais and off the dais, faculty members and dear students. It gives me a great pleasure to propose the vote of thanks. Our Honorable Vice Chancellor, Professor P.S. Edapatithaya, has been encouraging us in all our activities, and today he has presided over the function. I thank him for taking part in this inaugural function, for releasing the wildlife video. Thank you, sir. We are grateful to Professor Muralidhar L. Hegde for inaugurating this workshop and for his inaugural address. Thank you. I take this opportunity to thank Professor K.S. Jagannath Rao, Chief Guest of the function, for addressing the gathering in the virtual mode. Thank you, sir. I am grateful to members of Syndicate, Academic Council, chairpersons and faculty members of other departments for their cooperation and participation in this program. I thank Registrar, Finance Officer, Engineering Section and other staff for their support and the print media for their coverage of all our programs. Support rendered to us by the faculty members, research scholars and students of the department for organizing this workshop is gratefully acknowledged. Our students have taken a lot of efforts in designing the wildlife video and for the afternoon quiz, which I am sure deserves a big round of applause. I am grateful to the university for the financial support for this function. We have a good number of students and faculty members from different UG colleges and also the PG departments. I thank them for responding to our invitation. They have traveled all the way to participate in this workshop and to exchange ideas. I hope they will enjoy the deliberation and will have good interactions. I hope the time they spend here will be fruitful and will inspire them, which will serve the purpose of the workshop. I also thank Ms. Namita and Shamana, who have worked as the MC for this program. Thank you one and all.
Thank you so much, sir. Now it is time to capture this precious moment. It's time to have a group photo. I request our zoology department, that is nothing but Bhaskar sir, Shripada sir, Sham sir, NN sir, and all the zoology department to come on the stage to have a group photo. So please. We have Dr. Muralidhar L. Hegde with us to give a scientific session on the topic, a new era of DNA repair mechanism based therapeutics for aging and neurological disorders, brain health. So we are already aware of his achievements and we are really happy to hear those achievements, sir. So please come on the stage. The stage is yours.
Well, welcome back everyone. So I'm going to give a talk on a new era in DNA repair mechanisms based therapeutics in neurodegenerative diseases. So as we discussed very briefly in the morning, the initial about 4 to 5 decades was about understanding how DNA structure works, what is the predominant DNA structure in human cells and what are the alternative structures like zDNA and all, where both mine and Dr. Mushtaq's mentor, Jagannath Rao, was collaborating with Dr. Vishwamitra and made some very interesting breakthroughs, and they even found that some of the dementia-associated brains like Alzheimer's disease and other common dementia brains have more DNA converting into zDNA, like left-handed conformation and condensed form that affects the, how proteins are expressed in dementating brains and so on. So that was the, before the turn of the century. And in the first decade of the century, it was an era of DNA repair. What are the different DNA repair machineries in human cells? And in fact, at the outset, I would like to mention that, so our DNA gets continuously damaged. We breathe air, the oxygen itself produces byproduct like reactive oxygen species and other free radicals which damages DNA continuously. And in spite of that, and according to one estimate, a kind of broad estimate, it's about 10 to the 5 such damages occur in a single cell every day. So still we survive, and some of our cells live about 80 years like neurons. So that means that kind of huge DNA damage is still repaired very efficiently. And we think that this is still an underestimate because DNA is being damaged and continuously being repaired. It's very difficult to quantitate the exact amount of damage. You always underestimate that. So in spite of that, you have that such a huge amount of DNA being repaired and cells are living healthy. That means you have a very efficient repair mechanisms. And this was kind of undermined or ignored initially. The reason is there are more than one DNA repair pathways. As people started discovering DNA repair pathways, and I am talking about oxidative DNA damage, which is sometimes considered as mother of all damages. This is the more common damage which occurs under oxidative in aerobic organisms like us. So this kind of damages have, for each damage, you have at least 4 or 5 machineries or pathways. So there is a predominant pathway. If something happens to that, there is a backup pathway. There is a backup pathway for that. And other pathways can, co-opt for these pathways if there something happens for the primary 2 pathways. So for this reason, what happened was, people did knockout mice. Knocking out, as people discovered new DNA repair system, they knocked out that protein and found what is the effect of this, and they did not find any phenotype because there is a backup pathway which people did not discover that time. So that initially it happened for few repair machineries initially discovered like NEAL1, NEAL2, oxidative repair proteins and so on. So people thought that DNA repair is not that important because you don't show any phenotype if you knock out one repair machinery. So it's in fact other way around. So DNA repair is so important that you have 4 or 5 backup machineries to take care of the damage in one pathway does not work. That's the reason our cells survive so much longer. So more recently, we are talking about DNA damage. All DNA damage is not bad. And some of the DNA damage, because it occurs continuously, our body or cells utilize this DNA damage for some processes, even detoxification of some toxicants, or under stress conditions, cells utilize DNA damage to overcome that. And I will try to enlighten that in one of my projects. So now we are entering an era of DNA repair targeted therapies. So the backup pathways I mentioned, it's almost like, I call it as in the last couple of years, I'm trying to get a better understanding of this. So whenever there is DNA damage or any stress, our body is trying to recover that all the time. The first approach of a cell is to heal itself, trying to find a way to survive. And sometimes it invokes pathways which look pathological. Like, there could be some DNA damage occurring, it utilizes it to survive, and it looks pathological. And some of the treatments, particularly in the brain disorders, many of you know that, many of the treatments always comes in mouse models. People found this and it works in mice models, it works in monkeys or, other animals. They never, come to clinics, they never work in humans. The reason is, many of these repair pathways, they have backup pathways, one thing. And I was trying to mention that the body is trying to heal itself. What you see looks pathological, and people jump to treatment. For example, I will try to very briefly. I will try to as a background to my presentation, I would like to mention, but I will show data and explain more in depth. So whenever there is huge amount of a shock of DNA damage, like with radiation or shock of insult, cell tries to undergo immediately. Effect is trying to take rest. You don't want to be functioning and making more proteins, especially in brain cells. So it's called senescence. And senescence was maybe up to 2 to 3 years back was considered pathological. It's associated with aging. But under an acute stressor conditions, cells go into that dormant stage. And if you look at the a tissue, it looks like some 20% cells are not functioning, they are dormant. And if you try to activate them at that time, so they die. The reason they go under that dormant condition is they want to detoxify their, whatever pathological condition is occurring and recover and then start functioning. They don't want to start functioning when there is a toxin in the cell, so that, the proteins are, you make abnormal proteins. So many of the therapeutics, especially in the brain diseases, people which worked in animals were, because animal models are always acute models. You have very acute of condition of 50% of hippocampal cells damaged and so on, which does not occur in human conditions, which is more slow process. So our goal in our laboratory is trying to understand for these conditions how our cell or our body as a group of cells or a group of tissues are responding to a particular stress or particular protein toxicity, and trying to understand, and if they are protective, if those responses are trying to survive, you facilitate it. Sometimes they may stuck trying to get over some stressful condition. You facilitate the cells to naturally heal rather than prevent all those things cell is trying to do. So that's one of the theories which we are working on. So before starting my project, I would like to acknowledge members of our laboratory. Most of the work has been done by these people. So over the last 10 years, many of these people in the second row who worked in our lab and moved on, and some of them continued as postdocs. For example, like Velmarini Vasquez, who did PhD in our lab along with Erica. She came back as a postdoc. So Haibo Wang and Joy Mitra are more senior people who are working on two different aspects of motor neuron disease. They are like junior faculty. And Manohar and Vikas are postdocs. Vikas is in fact from Karnataka, from Kuvempu University. And Vincent Provosic is an MD PhD student. So we have a mix of basic scientists as well as kind of clinical students. So I am in a clinical department, neurosurgery. The chair of the department is a neurosurgeon. So it's like both translational work. And Pavana, who is my wife, who is here, she is our lab manager and senior research associate. So, and many of these people who worked in the last 5 years, they contributed immensely to the data I am going to present. And this is where we work. Mushtaq visited us about 3 years back, and we spent some time here. So this is Texas Medical Center. So now this is the largest medical center in the world. It is bigger than Boston now. So it has about 10 universities and about 20 different institutes kind of congregated in one area. So we have like 7 towers, and this is the research tower in Houston Methodist Research Institute, and these are the hospitals surrounded by hospital towers. And Houston Methodist is in fact one of the largest and best hospital system in Texas. Like you have for specialties like cancer, MD Anderson and so on, but overall clinical care, Houston Methodist is one of the best hospital systems. And since I did my PhD in 2005, I moved to University of Texas Medical Branch, Galveston, to pursue my postdoc, and that was with Shankar Mitra, as I mentioned. He was one of the world's leading authorities in DNA repair, like kind of fundamental aspects of DNA repair, and that gave me a lot of opportunity to learn basic aspects of DNA repair, which I am applying to brain diseases now. So I moved here in 2013, and in fact, this is one of the best campuses I felt that I will ever get to work with. Even the hospital, hospital is the primary priority, and research supports the hospital. And one example I give, this is a Methodist-based system, more service-based system. When patients go to hospital, for example, there is an outpatient procedure. I myself experienced that, and I was surprised actually. When there is an outpatient procedure, you are done, and pharmacy people, there are Walgreens and CVS pharmacy in the campus, and they come to your ward and take the prescription doctor gave and bring it to you. It looks so simple, but I felt that when you have a patient and you want to take the patient home, you don't have time to go to pharmacy and take medicine, or you cannot drop the patient home and come back, right? Some of these small things make Houston Methodist very attractive to patients. So it has a large pool of patients as well as donors. And even in fact, when we go for orientation, the first day of employment for anyone, so what they say is, for anyone, even they could be administrators, researchers or anything, the patient care is the first priority. So even if you have an important meeting or any engagement, so if you find on the way in the morning, some patient looking for something, you can take half an hour, 1 hour, and, make sure that you show the patient the way and then come back. It could be, even meeting with the president, you can, avoid that and, you should help the patient. This is the kind of service they give, and I myself was very much impressed and happy to be part of the system. And already mentioned briefly, coming to DNA repair, I'm sure Mushtaq as well, we are very privileged to be associated with some of the stalwarts in DNA repair, and unknowingly we learned a lot of things from them. Now if I look back, we actually learned a lot of things from them. Professor Vishwamitra, as I mentioned, he published about 14 Nature papers in about 10 years' time. And because of Jagannath Rao, our PhD mentor, we got to know him. We used to spend time in his lab. Mushtaq just mentioned that we used to go and sleep in his lab. It's in Bangalore, and we used to do PhD. So we used to go to his lab, and whenever we used to get 2 to 3 days time to do some circular dichroism and other experiments. We used to go and utilize it effectively, and we used to sleep in the lab itself, so that we can do the experiments early in the morning. So this was a great time during PhD. And Professor Jagannath Rao, as I mentioned, and you will hear, he will impress you in the second talk. So he is a visionary in science. He moves the field. So whenever there is a new direction, he gets so excited and gives you that opportunity. And he is one person in that time who highlighted DNA damage in aging and age-associated diseases, in collaboration with Professor Kalluri Subbarao and other people. And Professor Shankar Mitra, as I mentioned, he came here and gave a talk. He is one of the stalwarts in DNA repair. And these two people, Mushtaq mentioned in his initial address also, Lalji Singh and also Hargovind Khorana. I want to just remember their names on the occasion of this workshop. And, your Vice Chancellor mentioned very nicely that, our DNA and especially in the brain, is so complex. We don't even understand 20% of this. There are a lot of challenges to study DNA repair, or even brain itself, or DNA damage and its contribution to the brain. And the reason for these challenges is, you have animal models for many of them. You have liver cancer, you have, close to mimicking human condition, you have animal model, or, you can do the human tumor in animals. Even for diabetes and all, you have better, and they may not be perfect animal models, but better animal models. For human brain diseases, we still do not have perfect, animal models. The reason is these diseases start appearing or show symptoms maybe 30 years after actual disease happens. It's a combination of lot of comorbid conditions and age association that ultimately results in the disease. The aggressive animal models do not mimic that. They show the symptoms. You change the important factor, mutate a protein, they show the symptoms. But when you have to screen therapeutics, those therapeutics do not work because these animal models do not reflect those chronic accumulation of the diseased condition in humans. So for this reason, we do not understand, do not have the tools to study the disease in animals, and you cannot study the human disease except by imaging and other stuff. You cannot study the brain disease in live, any live humans. You have to use post-mortem brain, which has limitations. And one of the examples of those challenges I want to give you, this is one of the papers Mushtaq published. I am still very intrigued with this, we did not get the opportunity to pursue this because Mushtaq also moved on, and we need a lot of collaboration with clinicians and so on. One of the things, Mushtaq was during his PhD with collaboration with JSS and University of Mysore. Mushtaq was looking into this manic depressive disorders, bipolar disorder or unipolar disorder, where people have this wavy pattern of manic condition as well as depression, and a large percentage of them commit suicide. And it's very difficult to study suicide cases, if they have a fall or something, then difficult to do. But we took, one condition of, hanging, and people who hang to death, and we took some age-matched controls who met an accident and died, and we took their brain. And Mushtaq found very interestingly that the DNA is degraded in many parts of the brain in those people who committed suicide. I think we studied about 20 to 30 brains over a period of 3 years with Satyanarayan Rao, who is who provided those brains from Mysore. And very interestingly, we were very intrigued. Mushtaq did all the gels and DNA instability, melting and other patterns. From cerebellum, cortex, frontal cortex, midbrain you take, and it's a kind of a ladder pattern. You run in the gel, it's completely degraded. It almost looks like apoptotic DNA damage. But if you look at hippocampus, it was very intact. So we were initially, thinking maybe when people, hanged, the oxygen supply to the brain was, was not there, and maybe that led to the degradation later on. But if that is the case, hippocampal DNA should have been degraded, right? Only hippocampal DNA was not degraded. It's very interesting. So is it possible that you need memory? You need both short-term and long-term memory to commit suicide. Like you have to remember some bad things again and again. Maybe that's the reason your hippocampus is intact. It's almost like there may be some trigger. Like we were having a lot of intriguing discussion. It was difficult to pursue because I moved on to USA and Mushtaq. Also, we did not have research. But this is one of the challenges. I am talking about it. We were having the question of like, is it possible that when person commits suicide, the body gives a signal that it gives up? There may be other pathology, underlying pathology, which says that you cannot live longer. You cannot live longer happily or healthy. So, and then it is like apoptotic body death, where you have apoptosis in the apoptosis triggering in most parts of the brain. So this is one of the challenges I am talking about. To study this, you have an animal model, and you cannot have an animal model for bipolar disease, right? You cannot mimic that human condition. So some of these aspects are emerging again now with a lot of new technologies, tools, inducer pluripotent cells. You can make neurons and brain as a mini brain. So probably we can revisit many of these challenges which we could not address properly. We can come back. And for the students now getting into the field, I feel that next 10, 20 years will be the era of brain diseases, because none of the diseases have treatment. They have some treatment where you do brain stimulation and surviving cells will work a little harder and make you healthy for some time temporarily, but cells are dying. Those cells are, you don't have a treatment to prevent those cell death. So basically you don't have a cure for these diseases. And another reason why we don't have a cure is, you talk about motor neuron disease. I will show you in the next slide. So it's not one disease. You have a motor symptom, but there are about 20 different motor neuron diseases which shows the same motor symptom, but underlying pathology is completely different. And some may have overlapping pathologies. So what is happening pathologically is different in different diseases. Even we don't have tracers for PET scan and so on to identify what is the underlying protein pathology, unless you have a post-mortem brain, patient dies and you study the protein clearly. We don't even know what is the underlying pathology. So next 10 years, now the tools are emerging. This will be the era of brain research, and I'm sure we will make a huge impact on as far as treatment is concerned. So for students entering into the neuroscience, I think there is an exciting opportunity to study and to contribute to the new understanding of the brain in the next 10, 20 years. It was, it's probably same as, in the last 2 to 3 decades for cancer research and immunotherapy. It will be similar for brain research.